I'm going to go ahead and start my very short introduction. It is a pleasure to have Yimin Zou here from UC San Diego. He received his PhD at UC San Diego, showing how smart he is to pick such a fantastic place to go to the university. He then was a postdoc at UC San Francisco in Mark Tessier-Levine's lab, where he made very important observations about signaling pathways involved important in axon guidance, a long-term interest of Mark Tessier-Levine. He then became an assistant professor at the University of Chicago and was promoted and got tenure, and then in 2006 he moved to UC San Diego. He's a full professor there. He has served as chair of the neurobiology department at the main campus, not at the medical school. He's served on NIH study sections. It's where I really got to know him, and I'm delighted he's here because he just has amazing breadth and depth of knowledge about developmental neuroscience, spinal cord injury, axon regeneration, and axon guidance. He's the co-organizer of the next Gordon Conference on spinal cord injury, and I'm excited to hear us talk about Wnt signaling and its role in axon guidance and spinal cord injury. Welcome.
Thank you so much, Vince, for the invitation. Really excited to be here. Last time I was here, 2006, John invited me. I had a very good time. Give you some progress after the 13 years in between. So talk about axon guidance and spinal cord injury. I want to focus on what we have been doing with the Wnt signaling, but I wanted to zoom out a little bit about just one historical thing that the student might be interested in knowing. So as we all know, Ramon Cajal proposed this neuron doctrine. So in order for people to accept the neuron doctrine, he has to explain where the nerve fiber is coming from, because they're super long, they're well connected. So he had to show that. He went to the embryo to see when the nervous system is developing, and he started to notice that these nerve axons are, over time, if you fix them at different time points, they are growing longer and longer, and that's why he coined the word growth cone. So these images are one of those where he demonstrated that in development, actually nerve appear to be growing out of the cell. Of course, those are just images. A lot of people would already agree with him, but still, it's hard to understand. For example, in adulthood, when you have injury, and an axon regrow, and at that time the system is much bigger. So where do these new nerve fibers coming from? Are they the results of cell-cell fusion, or are they any result of any growth? So he, towards the very end of his career, he entered regeneration field, and this is one of the experiments where they did the sciatic nerve crush and then put a nerve transplant in there, graft in there, and then you can see that the nerve fibers are growing over time, and they actually grow into the graft. So on the left-hand side, he actually proposed the idea that in order for nerve to develop in a highly organized way, there must be chemical signals that guide them. So that's the axon guidance. So it turned out, then I realized that my axon guidance project over the years, one of the millions projects to sort of come from the idea that you need to actually demonstrate how the nerves connect themselves. But he proposed these there are chemical signals. But over here, when he was showing this data, there was no perturbation. But over here, he emphasized that, as you can see, these nerves seem to grow in certain direction. And he said, again, in the regeneration setting, there may be chemical signals there. And he even made a comment. He said, maybe some of these cues are the same molecules. He just mentioned that. But today, I want to tell you, our experiment actually, in a way, is consistent with his vision that Wnt signaling molecules actually are not only axon guidance molecules in development, they actually still regulate axon growth in adulthood. So this is actually part of the motivation. We wanted to understand how axons guide themselves into a target in development. But in reality, if it sounds like a very straightforward idea. It's probably true, but if you look at evidence, I'm proud to say the Wnt signaling is probably the only axon guidance system has been thoroughly studied in the context of regeneration, axon regrowth after injury, because there's many other examples of axon guidance molecules being induced after injury. But those functions haven't been really rigorously tested. It was still an idea. But I want to say for Wnt signaling, it's not an idea anymore. It's a fact that Wnt molecules will influence axon growth after injury. So I just wanted to start with that. So we, as I mentioned, when Ramon y Cajal was proposing this axon guidance idea, those were not really, there's no perturbation. But the experiment that came later in the 60s, this is probably one of the earliest experiments to show that there are guidance molecules in vivo. So over here in the salamander hindbrain, you have these methylene neurons that are growing from rostral to caudal direction. So if you cut a piece of the hindbrain and flip over 180 degrees and put it back, and those are the cutting pasting experimental embryology experiment. A bit earlier, that's how people discover Spinal organizer and the dorsal ventral patterning. A little bit later in the 60s, I think they ran out the early experiments and now moving to a little bit more later development stage. So this is like sort of a mid-gestation. When you do the cutting pasting for these animals, amazingly, they can still heal back and the tissue can still grow. So initially, you can see these axons would follow the original direction. But once they hit the border, they start to turn around and go back again. So this experiment shows that axons actually are trying to read environmental cues. Their direction is influenced by chemical signals outside. It's not like they know where they're going. So there are guidance molecules. So actually, they're suggesting there's a guidance molecule along the rostral caudal axis. So we became interested after I started my own lab. So when I was in Marc Tessier-Levin's lab, we were studying how these axons grow from the dorsal to ventral, how they don't grow back, how along this dorsal ventral direction. When I was leaving, actually, I was becoming interested in what happened to these axons after midline crossing because they turn anteriorly, rostrally. And at that time in the field of axon guidance, nothing was known about guidance cues along the anterior-posterior axis. Everything we learned about was midline crossing, midline dorsal ventral direction. So that's a totally new direction. And towards the end of my postdoc time, I begin to think about this question. When I was leaving, Marc Tessier-Levin really encouraged me. He said, you should work on that project because nobody knows a guidance molecule along this direction. And he was very generous. I took the project with me, and it turned out we have an opportunity to address the question, what are the guidance cues along the AP axis, because all the commissural neurons after midline crossing turn anteriorly. So we do a lot of experiments like that. For example, we can dissect the entire spinal cord, electroporated dorsal spinal cord to label a few neurons, and then we open the spinal cord and culture them. This is like a video that we took in Chicago a long time ago. As you can see, when axons cross the midline, they have this beautiful 90-degree turn to grow towards the anterior side of the nervous system. And then the guidance cue for that turning was not known for many years. And in fact, nothing was known about any guidance cue along this direction in any animal. So long story short, we found actually there's a Wnt gradient expressed in the floor plate. Actually 5 Wnts. This is one of them, the most abundant one, Wnt4, anterior high, posterior low. And these Wnt proteins can guide axons. If we put a cell aggregate secreting Wnt, you can rescue the phenotype. You can turn the axon rostrally or caudally, depending on where the molecule is there. So Wnt attract commissural axons after midline crossing to grow rostrally. And then 2 years later, we show that the descending axons, like we pick the corticospinal tract because it's growing in the opposite direction. And at around the time at P0, you can see a Wnt gradient, rostral caudal Wnt gradient, expression gradient. And in the dorsal funiculus, the CST axons are growing, and they're repelled by Wnts. They're repelled by Wnts using a receptor called the RYK. RYK is a receptor tyrosine kinase molecule, and then that pushes the axons down the spinal cord. When we inhibit the RYK by antibody injection, you can see the descending growth is blocked at the bottom one. So this is the antibody injected one. So very luckily, after our studies, many labs begin to show that Wnts also guide axons. So this is one example in 2006, Jan Garriga's lab at UC Berkeley, and together with Corey Bogman and Scott Clark, showed that in C. elegans, actually, Wnts also guide axons along the AP axis. So they're highly conserved. In this case, it's repulsion. So these mechanosensory neurons turn anteriorly after they reach the ventral point. In the absence of Wnt signaling, you can get the randomized growth of these axons along the AP axis. So very, very nicely, that's why I was able to get tenure relatively quickly, because Wnt signaling as a guidance molecule was was new at that time, and you need evidence. People don't know whether it's real or not, but turned out to be highly conserved. So we showed first along the AP axis, commissural neurons guided by Wnts, and also corticospinal guided by Wnts. And then in C. elegans, a few years later, in chick, also in a pattern. Yeah, so in chicken also was shown to be guided by Wnts. So chicken and fish later on. So the question is, which Wnt signaling pathway regulate axon guidance? So there are canonical, non-canonical signaling pathways. So over the years, we found that the components of the Wnt-PCP pathway are the molecules in the growth cone that are very likely directly influence axon turning. In a way, that kind of makes sense, because planar cell polarity is important for cell and tissue polarity, and it controls the polarity along the planar direction. So there are 6 components. They are localized asymmetrically. They interact across the cell asymmetrically. And we found the components of these PCP proteins are in the growth cones. And when you mutate them, for example, in the Van Gogh mutant, you can see it randomize the growth. In the flamingo mutant, this is the wild type. It's a conditional knockout. If you knock out flamingo from commissural neurons, you can see randomized the growth along the rostral caudal axis. Here is the frizzled. If the frizzled mutant, you can see randomized growth as opposed to control. More recently, we conditionally knock out Wntless. So Wntless is a cargo receptor for Wnt secretion. Without Wntless, no Wnt can be secreted. So if you delete Wntless from the floor plate using a Cree that is only expressed in the Cree, you can see on the left-hand side there's a control. In the conditional knockout, you can see the perfectly randomized growth. So over the years, we show that multiple components of PCP signaling pathway seem to be important for mediating AP guidance of the commissural neurons. So here's a summary. A few years ago, when I was writing this review, I was careful to ask the question whether PCP signaling components are important for growth cone signaling as a question mark. Looks like we can softly remove the question mark. Looks like the PCP components are going to be the key signaling mechanism for commissural neuron guidance in response to Wnt. In addition to commissural neuron, we also look at other parts of the nervous system. So in the midbrain, the dopaminergic neurons and the serotonergic neurons are also patterned along the AP axis. In the Y type, all the DA neurons grow rostrally. In the serotonergic neurons, there are a few of them grow rostrally, the others grow caudally. They're separated by the I think it's rhombomer 4 here. So there are Wnt gradients going up and down, these isthmus and rhombomer, and there are repulsion and attraction going on there. And we found that mutations of frizzle, flamingo, and Van Gogh, they all seem to randomize the growth of these axons along the AP axis. So not only in the spinal cord, in the midbrain, DA neurons, serotonergic neurons are also guided by Wnts along the AP axis. Here's one example. This is the Y-type DA neuron growing rostrally. In the frizzle mutant, you can see many axons grow in the wrong direction. So this is multiple neurons going to use this pathway. And very happy to report shortly after our publications, there are a number of papers from various animal systems to show that in the cases where Wnts are guiding axons, these PCP components all show the same phenotype as Wnt. So in the C. elegans system, the chick system, Esther, Stokely's lab, and also more recently zebrafish, frizzle, Wnt, frizzle, flamingo in zebrafish, Van Gogh, they all show the same phenotype in the zebrafish commissural neuron. So it's highly conserved. So definitely very reassuring that we are heading the right direction. So when these are happening, we became interested in the question about spinal cord injury. So these axons are happening to be along the rostral caudal axis, and they are definitely affected in spinal cord injury. And as we all know, that the spinal cord has circuits everywhere, and after injury, these neural circuits are often still quite intact. What is missing are the longitudinal connections along this direction. So if we can repair, we can encourage the growth of these neurons a little bit. That might help the function, connecting all the networks together through functional rehabilitation. There's going to be a very good. That's actually the reason for spontaneous recovery if people have some. But if we can encourage, that would be very nice. So we decided to. So we collaborated with Aussie Stewart to look at injury. So after so in adulthood, there's no expression of these Wnts in a normal spinal cord. But after injury, even 1 day after the spinal cord hemisection, you can see Wnts are diffusely increased surrounding the lesion, and Wnt1, Wnt5A, and Wnt4 is also highly induced. They are not induced in the core. Actually, they're absent in the core. And semaphorin was shown to be induced in the middle, but Wnts are induced outside. Why are they induced? They're probably going to be involved in part of the tissue repair system, because you need to, repair the wound, and you need to migrate the cells, you need to divide the fibroblasts and everything, all these cells. So you need to activate those pathways. So they're activated, but what is more important is that the receptors are also activated. So we found that RIC, the receptor that mediates axon repulsion, is activated on the axons that are injured, not the ones that are not injured. And if you look at this example here, in the P0, when the CST are growing down the spinal cord, RIC expression is pretty highly present in the ventralmost part of the dorsal funiculus. In adulthood, as you can see, the staining is quite weak. After injury, 1 day after injury, you can see the protein already induced. So here is the tracing from the BDA tracing from the motor cortex. If you look at the intact spinal cord, there's no RIC present, but this is 1 day after injury. You can see from the red and green, you can see a lot of yellow indicating that RIC is present in the injured fiber, on the injured fiber, and 7 days later, you can see a lot more. So at that time, we generated a polyclonal antibody against the RIC, which is the binding domain of Wnt. So this antibody can actually block repulsion. So over here, if we put Wnt1 in the cell aggregate, and you can typically repel CST axons in the explant assay, in the collagen assay. Wnt5A also have a very strong repulsion, but if we co-incubate the polyclonal antibody against RIC, you can see the repulsion is blocked. So that's very nice. And then we send this antibody to Xiaomin Xu's lab. So Xiaomin Xu's lab injected this antibody into the spinal cord. This is the control. This is the antibody injected. As you can see, after antibody injection, a lot of axons remain close to the lesion site, suggesting that the retraction of axon is reduced. And also, if you look at axon collaterals, there's also an increase of collateral growth. So over here, Xiaomin's lab did the unilateral labeling on this side, so only this side is labeled, and then the lesion epicenter is here. This time is the dorsal hemisection. As you can see, a big retraction, 1 millimeter above. There's very few axons left, but below the injury, there's no labeling. So the antibody-treated animals, if you look at the lesion site, 1 millimeter above, you can see a lot of axon remaining. But what is interesting is you see a lot of axon branches that are at the lesion site and in the intact tissue, and below the lesion site, some even grow across the midline. So suggesting that if you block the RIC signaling, Wnt-RIC signaling, you can actually reduce retraction, but also increase branch growth. So that was in 2008, and we actually the first time to show that any axon guidance molecules still function in adult nervous system after injury. So then we were in 2008, so we're very lucky, 2009, a very, very capable postdoc, a student from Mark Tazinski's lab, joined our lab as a postdoc. So he wanted to look at Wnt system a lot more rigorously. So he made a conditional RIC knockout mouse, and then he did this experiment by dorsal hemisection in a C5, and around that time, the foramen reach is becoming very popular, and it's a very good assay to show the CST function. Actually, without that assay, any other behavior, CST doesn't have a very big influence. So locomotion and others, CST's function is very limited. So even if you can have some anatomical improvement, the effect size is probably not going to be very huge. This is a very good assay because the CST is definitely required for the learning of this. This is a video that compared to what Vance is doing with in the mouse room, this is very primitive, but it's. I think the video is not working. As you know, they will reach out and yeah.
I'd just like to say it's not Vance doing that.
Sorry. Yeah.
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 Those images that you showed of the corticospinal tract,
Yeah.
with all the sprouting,
Yeah.
they're almost identical to what was shown in the early experiments with anti-no-go. And I also
Yeah.
can say that we did experiments with anti-no-go as well, and we found the same thing. So, is there an interplay between those two systems?
Yeah. So no-go. No-go is a myelin protein, and we typically don't call them axon guidance molecules. I think they have looked at whether no-go is involved in axon guidance. I don't see any clear, no study shows that it's an axon guidance molecule. So I think they're coming from a different source and may have a different function. One possibility is that the no-go might be regulating a different kind of set of neurons. So this could be, the different subset of neurons. Both are potentially very, very useful for regeneration. But this is sort of the Wnt signaling axon guidance axis. Yeah. So, but if you look at the fibers, they look the same, but they may not be the same cells. What happened to the force quit? This one? Oh, we cannot quit this. Anyway. Oh. Why is this? Okay, somehow I can see, you cannot see that. Okay, great. So this is the our result. So this is the learning curve for the foreign reach success rate, and after injury in the control you can see there's a baseline recovery, and then in the conditional knockout you can see a lot more, very significant increase of recovery. And if you look at anatomically, you can see the axon fibers in the conditional knockout is a lot more than the control, and there's even more axons here compared to here, but a big drop. Anyway, so this is just show that genetic evidence that the RIC in the CST actually is functioning as a receptor that senses Wnt as a repulsion. So if you remove RIC, you can get more axon growth and then there's functional recovery. So I'm not going to go into great details because it's been published, but I wanted to mention that we made a monoclonal antibody against the RIC. So now this antibody is going to block the binding between Wnt and the RIC. So this monoclonal antibody potentially can be developed into therapy. So we intrathecally infuse the antibody this time rather than injection. As you can see, we infused the antibody, and then this is the result with 4 months of experiments. You can see the antibody injected animals behave much better in the recovery. So this is very reassuring, suggesting that if you remove the RIC repulsion, you can have better growth. You can have recovery. But again, I want to emphasize it's not the repair of the original circuit. It's the reorganization of the connection, and it's sort of through neuroplasticity that reach the functional benefit. Encouraged by that, I think we really want to know, now that we just remove the repulsion, we can have more growth. What if we can increase the growth? Maybe we can do even better. So still motivated by that, we want to understand how Wnt actually guide axons. So today, actually, I'm going to talk more about axon guidance because I think understanding how axon guidance is regulated will provide more opportunities for us to intervene in the future. So as we were looking at all of these, so these are another continuously we wanted to, also I want to say that this is actually 1 year after our original publication in 2009, Yamashita's lab showed that in their own hand, injury, they did a contusion injury, and that induces Wnt RIC expression, and they made a RIC antibody also, infused the RIC antibody into the rat spinal cord. They get axon much better growth, and they did locomotion. They saw improvement of function. So they were able to replicate our study independently, potentially competing with us, but they were published a year later. And then after that, there are many other labs showing Wnt induction, and also seem to be like after injury, Wnt will be induced. So we are pretty confident with that. Okay, so what about understanding more signal transduction mechanism to try to further improve our ability to regrow axons? So I'm going to give you a little bit of a background about where we are at. And so this is like, we show that in the non-canonical Wnt signaling, we found a very interesting feedback loop. So it turned out Dishevelled-1 can inhibit Fzd, and Dishevelled-1 inhibit Fzd by causing Fzd hyperphosphorylation. When Fzd is hyperphosphorylated, it does not endocytose. That is antagonized by Vango. So Vango antagonizes that. When Vango is present, Fzd is less phosphorylated, Fzd can be endocytosed, and the signal can go forward. So what is important is that in the growth cone, so this is, okay. So as you can see, when you have Dishevelled-1, Fzd become hyperphosphorylated, and it's on the surface, come to the cell surface, and it's just the cell surface part, yeah. So if we put Fzd and the Dishevelled together in the growth cone, it's present on the plasma membrane. If we have Vango also together with Dishevelled, you can see Fzd come to the in the vesicles. So in addition to that, we also found that Dishevelleds are antagonistic as well. So Dishevelled-1 and 3 can cause Fzd hyperphosphorylation. Dishevelled-2 does not. And Dishevelled-1 and 3 can increase Fzd cell surface expression. This is the surface labeling, but Dishevelled-2 does not. Not only Dishevelled-2 is different from Dishevelled-1 and 3, Dishevelled-2 actually inhibit Dishevelled-1 and 3. So if you have more and more Dishevelled titrated in, Fzd phosphorylation is reduced. So Dishevelled-1 induced Fzd phosphorylation is reduced. So there's a lot of antagonism going on. So these are sort of biochemical experiments, and these allow us to propose ideas that maybe there's a lot of antagonism going on. You have Fzd, Dishevelled-1 antagonism, Dishevelled-1, 2 antagonism, and those signaling antagonism could be important for setting up some kind of a difference. If these molecules are localized differently, that can create asymmetry. So one of the things we noticed is that in the growing Phylopodia, the tip of the growing Phylopodia has a lot of Vango. And the Phylopodia that are shrinking have very little Vango, so suggesting that some of the Phylopodia get selected because Vango is present at high level, and that increases the Fzd endocytosis and then promotes the actin polymerization. The other Phylopodia has less Vango, so PCP signaling might be used to select different Phylopodia. This is how Polarity was used. So that was just one of the ideas. So if we put the growth cone in the Wnt gradient, you can see the growth cone, the side of the growth cone that is seeing more Wnt will have more endocytosis than that. So we think that little difference might be somehow amplified by this kind of, a lot of this antagonism. For example, if you have a bit more Wnt signaling here, then you have more Fzd endocytosis, which activate Dishevelled-2. These Dishevelled-2 in these Phylopodia might inhibit Dishevelled-1. So this Phylopodia Dishevelled-1 is inhibited, and then inhibit Dishevelled-1 allow more endocytosis of Fzd, and that could be an amplification because more Dishevelled-2 will inactivate more Dishevelled-1, which will activate more Dishevelled-2. So these are the hypotheses now. We think that somehow the asymmetry is created by this kind of a dynamic movement of endo and exocytosis, and the gradient initially might trigger a small difference that may be amplified by a few biochemical amplifications. So this is hypothesis. These are results sort of there, and so from here to here, it doesn't, this hypothesis may not be right, but that's the kind of hypothesis we have. But the reason I want to mention that is that after what we showed here, 2 years later, and another year, 2 years later, there are 2 other papers that supporting our hypothesis. So in C. elegans, actually, Dishevelled also has 2 functions. One is to promote PCP signaling. The other one is to inhibit PCP signaling in axon guidance. So this is work done by Marty Chaufey's lab in New York. And they found that the Fzd is inhibited by Dishevelled by phosphorylation, the same as what we found. And then Vango antagonized Dishevelled and reduce Fzd phosphorylation. So what we found in mammalian system, they can completely replicate in C. elegans using genetics. Another lab showed later on that the Vango promote Fzd endocytosis. So what I'm confident about is that the biochemistry that we discovered seem to be conserved. And how are they exactly used for planar cell polarity signaling? We still are working on it. It will probably take a few years, but I think that it's important to recognize that it's not a linear system. It's a lot of components. They are asymmetrically localized. They antagonize each other, and there may be a sequence of those action, and there's maybe and then there's an amplification of the signal. So we might have understood some of the biochemistry that we showed here, and very happily other people showing the same thing, that these are going to be our substrate for our next step. We don't have the answer yet, but it feels like this is this should be a place where we should be looking for answers. How does asymmetric signal occurs in the growth cone when the growth cone seeing a concentration gradient, and that kind of amplification might be essential for growth cone guidance, because all of the guidance cues are very shallow, the gradients are very, very shallow. There's going to be some kind of a way to amplify the signal. So this could be in the next few years, we're going to continuously use this system to understand how axons are guided, how axons grow. And so today I'm going to give you a little bit of a progress on that. That we don't know how to immediately how to apply that to spinal cord injury, but it's sort of the progress along that direction of understanding growth cone guidance. So the other reason I introduced all of this background to you is I wanted to mention, because that's set of the background for my next talk. So as you can see, phosphorylation controls localization and also endocytosis, and that seemed to be critical for the whole process. And in fact, other people showed in PCP signaling, endocytosis is absolutely essential for planar cell polarity signaling. So we become very interested in understanding how phosphorylation is regulated. So one way to do that is to figure out how physal is phosphorylated, and then you can control the dynamics. So it turned out that we discovered a physal kinase. It's very surprising. For many years, we've been looking for physal kinases. They all turn out to be inhibitory to phosphorylation. So we found a kinase. The kinase seemed to matter for guidance, but when we look at the physal, they all inhibit phosphorylation. This time we found a kinase seemed to be directly phosphorylate physal. So this a post I was looking for the kinases. So it turned out the physal has, we know by then, at least 7 phosphorylation sites. They're all phosphorylated by different kinases. So if you look at all the sequence, you do some prediction, there are many, many candidates. We don't know which one to start with. So one day he realized that all of these protein kinases are actually can bind to one protein called LOC2. So LOC2 is a very big protein, and itself is a kinase, and has a lot of other protein which can binds to, and these are a lot of kinases, and they can also phosphorylate. So Kisuke wanted to test whether LOC2 regulates physal phosphorylation. So he first of all, he tried to knock down LOC2 using shRNA. So he did 2 shRNA construct, and they both knocked down LOC2. So when he did that, he found that the Dishevelled induced physal phosphorylation is reduced by these 2 shRNA. Very, very nice. So then the next thing he did, he bought some LOC2 enzyme. You can buy these enzymes, recombinant protein, and he made the cytoplasmic region of physal, put them together, did in vitro kinase assay. For sure, this is the phospho tag, which is help you to separate the phosphorus species of the protein. You can see it's phosphorylated, and you can see there's a phosphorylation band here that is increased in the mutation. So this is the this is the phosphorylation here, but it's a Y type. And then GS is a gain of function mutation of LOC2, which has more kinase activity, additional phosphorylation, and then kinase dead. These 2 bands are gone. So he did a mass spec and found actually there's a site which is additional site. So this is a T598. LOC2 can directly phosphorylate physal on this particular site. And then so this suggests that the physal, one of the kinases could be LOC2. That's very interesting. And then so this is over here when he overexpressed LOC2. You can see so this is a total fraction. And the phosphorylation is increased in the Y type. Phosphorylation is increased in the gain of function. Phosphorylation is also increased in the kinase dead version of LOC2. So that means the kinase domain of LOC2 is not required for all of the phosphorylation. So suggesting the other kinases are still being recruited by LOC2. And on the cell surface is the same thing. Dishevelled 1 can increase physal cell surface localization, and the Y type LOC2 can increase that, and then gain of function increase that, and kinase dead is also doing that. So what Kisuke think is that LOCK2 phosphorylates Fzd directly on this side, but uses other kinases to phosphorylate the other sides. So at least we found a kinase that can regulate Fzd phosphorylation. That's a very important tool for us, and we don't know what the other kinases are. So then we can begin to test the function of LOCK2 in axon guidance. So Kiski went ahead and made knockout mice using CRISPR. So he knocked out using CRISPR, he knocked out LOCK1 and LOCK2, both because they're kind of redundant. And of course, after CRISPR, we back cross 6 generations back cross to wild type, just try to avoid potential off target. So we're very confident these are clean results. And when he knock out more and more LOCK, you can see in vivo, Fzd phosphorylation is decreasing with more and more removal of the copies of LOCK1 and 2, suggesting in vivo it is very likely, if it's not a direct kinase, it will be regulating Fzd phosphorylation. That's the kind of thing we wanted to find out. And then what about the phenotype? So Kiski decided to use a new way to label commissural neurons. So we typically use dye-eye tracing. That we and Esther, we do a lot of that kind of dye-eye tracing these neurons. It's very hard because when you label the neurons, you don't know which one's going to be labeled and require a lot more consistent training. So Kiski created a line that it will be easier to visualize. So in this case, we used the Math1 promoter. So these commissural neurons, they are commissural neurons. They grow across the midline, but after midline crossing, they immediately turn rostrally. But they grow at an angle towards the brain. So that way you can see from the angle where they're going. If everybody's going very close to the floor plate, if you image them, they're all going to be very bright over here. You don't know the direction, but if they have an angle, then you can see we're turning this way, turning that way. And if we do sparse labeling, you can see axons better. So this is a cleared spinal cord with this labeling, so Math1 Cree, and then TD tomato label. You can see all of these axons growing eventually, and across the midline, after midline crossing, they all grow anteriorly. So that way you can label them, but this is a sparse labeling, so you don't label all of the neurons. You can see the axons relatively clearly, similar to the approach that Kevin took for the optic nerve, the regrowing optic nerve. So with that, Kiski noticed that there's a pretty strong phenotype in this commissural neuron. So on the top are the controls. You can see nice crossing and then turning rostrally, but in the Y-type control, you see the axons highly organized. But in the LOCK1, LOCK2 condition, regular knockout or double knockout, you can see a lot of axon guidance defects. Most strikingly, there are a lot of AP guidance defects, but most strikingly a disorganization. As compared to the Y-type, these axons are growing very organized. Seem to be in a group, very organized turning, but in the mutants, they're turning in the wrong directions. They're wandering around, but they don't seem to grow together. They seem to lose the communication among them. So again, there's another example. You can see very organized growth after midline crossing, but in the LOCK2 knockout, there's a lot of descending growth, but more interestingly, there's a huge disorganization among these. So this phenotype is interesting. It does have AP guidance defects, but it's not as strong as you knock out Fzd. It's probably not surprising because Fzd is the key receptor. This is one of the kinases. So this kinase might be doing something in perhaps enhancing the signal or maybe something else. So I just want to give you one more example about our new results on Dishevelled. So remember I mentioned Dishevelled 1, 3 are redundant. So if you knock out Dishevelled 1, axons still grow properly. Dishevelled 3 knockout is still normal. But if you knock out Dishevelled 1, 3 together, you can see the randomized growth. Here is the labeling of that. On the left-hand side is the control. You can see they're growing across the midline. After crossing, they all grow anteriorly at an angle, so you can see very clear anterior growth. This is a double knockout. Double knockout, they cross the midline, they grow randomly, going up and going down. As you can appreciate, this type of neuron allow us to see the turning angle much, much better. This is a double knockout. So the AP guidance defect is a lot stronger than what we see in the LOCK2 mutant. LOCK2 mutant has AP guidance defects, but most strikingly are the disorganization of that. So here is the conditional knockout Dishevelled2 itself. You can see the conditional knockout. You can see the AP guidance defects is a lot stronger than Dishevelled and then LOCK2 mutants, where you do have AP guidance defects, but what you see a lot is the disorganization of these axon growth. So we become fascinated by that because this suggests that axons might be typically they might talk to each other when they're turning. So when you look at the commissural neurons, there are millions of them crossing the midline, and then the midline crossing is very slow. It takes 8 hours to cross the midline, and after crossing, it takes another 1.5 hours to turn. So there's a lot of interaction time. So we asked the question whether the growth cones actually might talk to each other during this time and then organize themselves and turn as a group. This is interesting because in planar cell polarity, the community effect is absolutely essential. So these cells, they are polarizing themselves, in the hair, Wnt epithelium, for example. They talk to each other. There's a lot of non-autonomous interaction, and the entire sheet of epithelial cells polarize themselves. It's not individual cells. They have their own signaling within the cells, but the communication among the cells are essential for the polarization. So if you look at the commissural neurons when they're turning, it's very similar to planar polarity. The whole group of growth cones might actually communicate with themselves and turn together as a group. So this is something we wanted to entertain. Now, we also got a hold of the gain-of-function mouse. The gain-of-function mouse actually, we are not allowed to cross with other markers, so we use the dye tracing. We also saw very similar disorganization. We have AP guidance defects, but again, mostly you can see a lot of phenotype of axon not growing like very nice angles. You can see they're going up and down. They seem to be disorganized. So first, we want to test whether the growth cones do interact with each other, and if the growth cones interact with each other, are they regulated by LARC, for example? So here, these two graduate students, they did this experiment by looking at how growth cones might interact with each other using live imaging, and then look at the mutants. And they found actually, interestingly, that they found on the left-hand side there are examples of control where the two growth cones, when they touch each other, they seem to be, they join together and they form, they get together immediately and they grow alongside each other. Whereas in controls, they see a lot of examples where growth cones would touch each other and wouldn't like to fuse. So here is like touching and testing, but they get together and then they separate. It's not like in the Y type when they get together, they immediately fuse and grow together, suggesting the interaction is different. We don't know what it means compared to in vivo. So that's the Y type versus knockout. And then the next slide will show the gain of function. So on the left-hand side will be examples of these growth cones. Once they see each other, they immediately get together and axon grow along each other. Here's another one coming, maybe. So this is a gain of function where you have too much phosphorylation. You see the same thing. So this axon is going to try to touch this growth cone, and they don't fuse. They go testing again. So here's the quantification. The next slide shows the quantification. So basically, on the top, it shows that when the blue and the red axons touch each other, they fuse, they merge together, they continuously stay together. Whereas in the knockout, they will touch and then they separate. And they touch again and separate. They don't seem to fuse that readily. So we think that the communication is interfered by both hypophosphorylation and hyperphosphorylation of Fzd3. So I just want to mention that the community effect is very essential for planar polarity signaling. Within the cell, these components antagonize each other, but across the cell, they form asymmetric cell-cell junction. These interactions are absolutely important for the establishment of cell polarity. So we think that something like that maybe happen in the growth cone. That could be a very, very good way for growth cone to use a relatively shallow gradient and to turn together as a whole sheet of growth cone. They can sample a more reliable difference of gradient. So we did a little bit more biochemistry to test this idea. So to mimic the cell-cell interaction, we first transfect the cell with Fzd3 and the flamingo. on one cell, and then the next cell we transfect with flamingo and Van Gogh, and then we culture them separately by 1 day, and then we put them together to see whether the Fzd and the Van Gogh interaction across the cell can be affected. So you use the Fzd, for example, antibody to pull down the complex and to see whether Van Gogh is there. So what we found is that so if you put Fzd only, the Van Gogh only, there's some interaction there. But if you put flamingo on next to Fzd, there's an increase. If you put flamingo next to Van Gogh, there's not so much increase. But if you put flamingo on both sides, you can see a much bigger increase. So suggesting the flamingo, which is a adhesion GPCR, they have cadherin repeats, is a protein that bring the complex together to increase the chance of interaction between Fzd and the Van Gogh. Okay, so that's interesting, suggesting there is a complexes in that's how they potentially work together. And then if you mutated the site of this phosphorylation site by log2, interestingly we see a greater interaction. So when this site is mutated, the interaction between Fzd and Van Gogh is increased. If you mutate the other sites, it doesn't affect interaction. But if you mutated all of the sites, then again you see the interaction is increased. So suggesting that during the growth cone turning, you need to have a fine balance of Fzd phosphorylation, because Fzd phosphorylation brings Fzd to the plasma membrane, and then the interaction with Van Gogh is then regulated by log phosphorylation. So when log is phosphorylated in the site, it weakens the interaction. So the fine balance of Fzd phosphorylation is essential for the proper interaction of growth cone, and having too little phosphorylation, too much phosphorylation Fzd will cause the disorganization of the turning and also affect the growth cone interaction. So we want to show additional evidence of that kind of interaction. So this is a conditional knockout when we knock out all of the flamingo using this Cre line, the Wnt1 Cre, which will be deleting flamingo from all commissural neurons. As you can see, the axons grow randomly along the AP axis. So we decided to knock out flamingo in individual neurons to see whether because of the community effect, if this growth cone doesn't have flamingo, the other growth cones have the flamingo and the other components are there. What if this growth cone can use the neighbor information to turn? So we want to delete flamingo from one individual growth cone, do the mosaic kind of genetics, and to see whether axons are affected. So for doing that, we have this Math1 Cre, Wnt1 Cre ERT2. So over here we use low tamoxifen to introduce sparse knockout. So this is the midline. So the red line are the markers or the TD tomato showing the recombination. As you can see, a lot of the dark area, because these are the Y-type neurons, only the individual axons have been deleted the cells of 3. And when we did that, this is the control. You can see in the sparse knockout, they all turn correctly. So that means if the surrounding neurons have flamingo, that neuron actually do not need flamingo. They can use the other neighbor information to turn correctly. Here is the quantification of that. We did many animals and show that there's no difference actually compared to Y-type and the conditional knockout. So if you don't have flamingo in individual axons, they can still turn correctly. Here's another example, very hard to get. So if you sometime when you inject low tamoxifen, it's actually not low enough, so it's still kind of high. But in the animals, as you know, the rostral animal developed earlier than the caudal animal. There are more axons in the rostral side. So in the same animal, rostral, caudal, in this particular case that we see guidance defects on the rostral side, but not the caudal side. So that means when you delete cells in enough neurons, you can begin to see that. So I just wanted to say that we now have evidence that axons, growth cones might talk to each other. So for axon guidance, axon-axon interaction has been known for many years, very important. There are many examples. For example, fasciculation is super important for axon growth and guidance. There's also selective fasciculation, and defasciculation, and also an arborization. But all of those are found to be interactions among axons or growth cone with axons. We haven't really looked at issue whether the growth cones might talk to each other. So this is the first case where we can see that in the growth cone, we propose that the growth cone can turn as a community, and the phylopodia interact with each other, those kind of interaction might be a way to amplify the signal, just like what is done in planar cell polarity. Okay, so this is something, it's a new concept, and it has been very difficult to publish. We have reviewers are super excited about this conceptually new idea, but there are other reviewers want exact evidence of how they really interact. So the best evidence we could have was the in vitro culture interaction, but that's honestly not exactly the same as, so we couldn't come with all the evidence, but this is a new concept we're trying to entertain, but hopefully we will have better experiments down the road. But I really believe that the growth cones will need to talk to each other to be more efficiently pathfinding. If you think about axon wiring in our brain, they're so highly organized, and then if they are all explained by Natrian gradient or Wnt gradient, it's so hard to use those gradients to build these networks. I think the neurons have their own interactions. These interactions can self-organize to quite a bit. When it comes to turning, to be able to turn so precisely together, it's not just the Wnt gradient that's doing. The Wnt gradient is just giving a north-south general direction. The growth cones actually know how to organize themselves. We need to emphasize this aspect of wiring to really understand if we want to repair the brain, we should be able to activate those kind of mechanisms that the cells have in addition to guidance cues. Just wanted to emphasize that hopefully potential will be useful. So obviously, LOCK2 is very important for Parkinson's disease. So I just want to mention that earlier we showed that DA neurons are guided by Wnt and PCP signaling. So this slide shows that if we knock out Fzd3, dopaminergic axons will grow in the wrong direction. So we decided to look at lock mutants, look at the axons in the lock mutants. So it turned out actually we saw similar defects, but again, much weaker than Fzd3. So it's not surprisingly weaker. So in the LOCK2 mutant, the Y type, LOCK2 knockout, you can see a few axons grow in the wrong direction, and axon become disorganized. So the bundle is not that tight. And in the gain of function, this Y type, the gain of function, you see more axons going in the wrong direction. So really suggesting that DA neurons are also using LOCK2 as a Fzd3 kinase for precise wiring of the DA neuron. Okay, so this is my summary. So LOCK2 promotes, so the new result shows LOCK2 promotes Fzd3 hyperphosphorylation by directly phosphorylating Fzd3 at this site and also by recruiting other kinases. And LOCK2 is required for precise AP guidance after midline crossing. And this site weakens the interaction, and a proper level of LOCK2 kinase activity is required for normal growth cone interaction, which we think this interaction is essential for the organized concerted turning of these axons. And interestingly, in the midbrain, dopaminergic axon guidance also require lock, proper lock signaling in development. So just wanted to thank all of the students, postdoc who did the work on axon guidance, spinal cord injury, and the recent lock work is done by Keisuke Onishi. Thank you very much.
Great talk.
Yeah.
Is there more LOCK2 in growth cones relative to other places and relative to other kinases? Because canonical Wnt, you have, I'm sure that those kinases are expressed in the growth cone as well. Why is it the PCP pathway is dominant?
Okay, so canonical pathway, we actually have evidence that if we knock out beta-catenin, they can still turn correctly to the rostral end. There are some differences in the cell body position, and also the axon growth rate is reduced in the canonical knockout, and the DCC level was reduced, but anterior turning was normal. I think canonical pathway is a pathway that controls gene expression, and it regulates cell differentiation, survival, and it's very important. But for directional, the local growth cone function, planar polarity molecules are in the growth cone. We think these are the molecules who are directly polarizing the growth cone in response to Wnt gradient. So for the particular turning, that directionality, canonical PCP pathway seem to be the main player for that direction. Okay. We didn't see any difference of gene expression. So one possibility is that the canonical pathway regulates the expression of PCP genes, so make it accessible. But when we knock out beta-catenin, PCP signal is still normal. PCP proteins are still there. So they are interesting, the canonical, non-canonical pathway, they can function simultaneously. And I think they can do different things at the same time. Sometimes they talk to each other. There are examples where PCP signaling or non-canonical signaling can inhibit canonical signaling. But there hasn't been a lot of evidence of canonical pathway regulating PCP. So, yeah. And we also look at sonic hedgehog. So sonic hedgehog, if you mutate smoothened, axons grow randomly along the AP axis as well. In the hedgehog mutant, we found that PCP gene expression not affected, but one molecule is upregulated in the hedgehog knockout. That molecule is called SHISA. So SHISA is a protein that in the ER that binds to Fzd and inhibit Fzd glycosylation. So when SHISA is knocked out, Fzd can be glycosylated and come to the cell surface. So we found actually hedgehog regulate the sensitivity of PCP signaling. So by having SHISA, Fzd is not on the cell surface. It's staying because it has not glycosylated. But after midline crossing, commissural neurons see enough hedgehog signaling, and then SHISA is downregulated. Fzd come to the cell surface, and they're turning rostrally. So we show that if we overexpress SHISA, after midline crossing, axons grow randomly, because Fzd never come out. But if we knock down SHISA, axons can turn rostrally before midline crossing. So SHISA is one of those switch that control the timing of Wnt response. Only after midline crossing, commissural axons become attracted by Wnt because of the regulation of SHISA through hedgehog signaling. So it was not a direct regulation of PCP components, but rather a molecule that regulates Fzd glycosylation. So we showed that, we published that 2 years ago. Yeah.
This is just kind of a general question about repairing the spinal cord.
Mm-hmm.
But in watching
Yeah.
the complexity of the interactions that
Mm-hmm.
you showed us, thinking about things that promote regeneration, like
Mhm.
kinase
Mhm.
inhibition,
Mm-hmm.
and DC current electrical fields, what I want to ask you is the relative importance of getting an axon to grow
Mm-hmm.
versus getting an axon to create a new circuit.
Mm-hmm. Mm-hmm.
Like I'm wondering if some of these
Yeah.
things that will get an axon to grow may fail because it doesn't form a new circuit.
That's definitely, I think it has been recognized. So if you have growth and then the axon not going to the right place, it may not help. And actually, sometimes too much growth might be even worse because it disrupts the normal connection even better. I think, but in the meantime, also when you have some growth without any sort of guidance, you can still gain function back. There's a lot of examples like that. So I think even if when you're not interfering, the growth cones, the axons may be recognizing the signal that you don't know about. So when they're growing back, they know where to grow back. So for typically in the phantom limb syndrome, it's very characteristic where the reorganization has a certain pattern. So they're recognizing something. So in cases where you get good function recovery, maybe those cues are present in a way that allow a functional connection or allow a new connection that can be trained to get this function. But there could be other cases where those things are not available, so you don't get recovery. Or even worse, when you have a lot of axons there, it's going to really mask the normal signal. So it is very sophisticated, but I think knowing how axons are regulated will enrich our understanding of these different cases where sometimes we can get some function back, not the others. Maybe there are cues there helping them, and then the other cases where it's not possible, it's not allowing the connection to be there. But again, when you I think it's typically very difficult to repair the original circuit going back, but potentially more doable by growing axons and potentially making more functional, sort of a new circuit. But over there, I think, if you know how to organize the axons better and with functional guidance, that's why the rehab function is so important to mature the circuit, because when you have these new connections, they're not functionally meaningful. It's just like development again. It takes so long for the neural circuit to mature, to carry function, but the repaired axon also need that time to be trained. And those knowledge about axon guidance, synapse formation will be handy when it comes. So that's why we are continuing with this direction. Yeah, but it's very exciting to recognize that it's not that simple. Just growing axon may not be sufficient. Thank you.